This method permits culturing Mycobacterium tuberculosis under strict low iron conditions and purifying membrane vesicles from the low iron culture filtrate. Because iron limitation is known to stimulate membrane vesicle production, this protocol yields high abundance of pure membrane vesicles that can be utilized for further biochemical or functional assays.
We will demonstrate the protocol using the Mycobacterium smegmatis, the non-virulent strain. This protocol can be followed in BSL-3 facilities using Mycobacterium tuberculosis, the virulent strain.
To begin, prepare 1 liter of minimal medium by dissolving 5 grams of monopotassium phosphate, 5 grams of L-asparagine, 20 milliliters of glycerol, and 2 grams of dextrose in 900 milliliters of deionized water in a plastic container. Adjust the pH to 6.8 with 5 normal sodium hydroxide, and adjust the volume to 1 liter with deionized water. Then add 50 grams of metal chelating resin and gently agitate using a magnetic stir bar for 24 hours at 4 degrees Celsius. The next day, stop the stirring and let the resin sediment for about 30 minutes. Sterilize and remove the remaining resin from the solution by filtration through a 22-micron filter unit with a plastic receiver. Now, supplement minimal medium with 0.5 milligrams per liter of zinc chloride, 40 milligrams per liter of magnesium sulfate, and 0.1 milligrams per liter of manganese sulfate.